Hi, and I will introduce myself quickly. My name is Kevin Wang, and I am a senior bioinformatics scientist at Illumina. And it's my pleasure to be chairing this section. And our first speaker of the session is someone who has very, very similar name as myself, Kevin Wang, and he's from the University of Melbourne, I believe. So take it away, Kevin, when you're ready.
Yep. Can you see my slides?
Yes.
Yes.
Okay,
Yes. Perfect.
yes.
Yep.
And I'll give you a minute around, maybe towards the last minute.
Yep. Thank you. All right. Hi, everyone. My name is Kevin, and I hope you're all having a lovely day today. And today I'll be discussing my research project concerning the associations between vitamin D deficiency in late infancy and the consequent outcomes on food allergy in early childhood. In particular, a secondary data analysis was conducted on the first 3 waves of the Health Nuts longitudinal study cohort. This project was completed under the supervision of Dr. Daisy Shepherd and Associate Professor Rachel Peters, both of whom are based at the Murdoch Children's Research Institute, or the MCRI. I'd also like to add that this project was completed as part of a capstone research subject during my Master of Biostatistics degree at the University of Melbourne. Our project consisted of 2 research questions, the first of which being whether vitamin D deficiency, or VDD for short, at age 12 months may be associated with the persistency of food allergy by age 6 years. In contrast, however, our second research question was concerned with whether a resolution of VDD corresponded with a resolution in food allergy status. For the purposes of today's presentation, only the first research question will be discussed, whereby a causal analysis was performed. For both analyses, we've considered only peanut and egg allergies due to their high prevalence within our dataset. But first, why food allergy? Well, it may come as a surprise to some, but food allergy is currently a significant public health concern, with evidence over the past 2 decades indicating a growing prevalence with no signs of halting. Unfortunately, however, there is currently no curative treatment aside from simply avoiding the allergen of concern. Of interest, Australia is now infamously known as the food allergy capital of the world, with an estimated 10% of 12-month-old infants being affected by food allergy. Given this increasing prevalence, as well as the fact that the greatest burden is on preschool-aged children, it is clear that there is much research to be done. Over the past decade or so, one of the risk factors identified is that of an individual's vitamin D levels. In particular, some evidence have revealed evidence of its deficiency being a key risk factor. Unfortunately, however, results remain mixed and the role of vitamin D is still not yet well elucidated. Nonetheless, this is an emerging field with only 1 study so far having examined an association between VDD and the persistence of egg allergy. I would now like to discuss a methodology underpinning the research that we conducted. So, Health Nuts is an ongoing longitudinal population-based cohort study conducted by the MCRI, and it consists of 5,276 participants who are recruited from local government-led immunisation clinics in Melbourne whilst attending their routine 12-month immunisations. A vast amount of data was collected from parents via questionnaire, and infants were assessed for food sensitisation using skin prick testing. Those found to be sensitised were then further invited to attend a clinical assessment at the Royal Children's Hospital, whereby they were formally diagnosed with food allergy using an oral food challenge. For the second wave of the study at age 4, participants were followed up again by questionnaire, with those who had attended the baseline assessment once again invited for another clinical visit. For the third wave of the study at age 6, all of the participants were invited to either attend the hospital clinic or to have a home visit by a study nurse. Now, in the present context, our exposure of interest would be vitamin D deficiency at age 12 months, and our outcomes of interest would be the persistency of either peanut or egg allergy by age 6 years. So how do we define these variables? Using the blood samples collected at baseline and follow-up, researchers were able to calculate the participants' 25-hydroxy vitamin D3 levels, this being the primary storage form of vitamin D in the human body. These blood serum concentrations were then categorized into a binary variable relating to deficiency, whereby the clinically relevant value of 50 nanomoles per liter was used. Now, the persistency of a food allergy is quite self-explanatory, I'd imagine, and two binary variables were also generated for these food allergies. The directed acyclic graph, or DAG, was constructed using a combination of evidence from previous studies, as well as input from the content expert. Of note are those covariates denoted in red, whereby these were identified a priori to be the common cause confounders to be adjusted for in our primary analysis, these being maternal place of birth, season of birth, socioeconomic status, and maternal vitamin D supplementation use during pregnancy. Our designated model for evaluating the association of interest was a multiple logistic regression model. This model is applicable as our outcome has been aggregated into a binary variable, and it is multiple in the sense that we've included a number of independent variables for adjustment. I would like to add that although the assessment of food allergy had occurred in waves, our outcome variables are only derived from this information and hence do not maintain a longitudinal data structure themselves. Presented on the screen here is the logistic regression equation on the log odds scale for the sake of clarity. P hat would be the estimated probability of a persistent egg or peanut allergy based on our data. Hence, for a given participant I, log of P over 1 minus P would be the log odds of a persistent allergy, and it can be expressed as a linear combination of coefficients and subject covariate values. The betas in this equation would therefore be the fixed and estimable parameters of interest, whereas the x's would represent an individual's values for a corresponding covariate. Beta 0 would therefore be the baseline log odds of a persistent allergy, assuming all covariates are held constant at their reference level. If we were to let beta 1 represent the parameter pertaining to VDD status, then our measure of interest would be the odds ratio comparing persistent allergy, comparing those with to those without VDD at baseline, and this would be obtained by exponentiating our estimated value for beta 1. A number of sensitivity analyses were also performed, however these will not be discussed in great detail today. These included an extended set of covariates for adjustment, consideration of vitamin D levels on the continuous scale, as well as consideration of vitamin D in quintiles, which I'll touch on briefly. Moving on to the results and discussion. The table presented here includes estimates of the odds ratios when fitting our model to our data, specifically for egg allergy. As we can see, the point estimates of the odds ratios are below 1, the null value of no association. Hence, when considering our data, this estimate would suggest that VDD at age 12 months might in fact be protective against the persistency of egg allergy by age 6 years. When considering our 95% confidence interval, however, we can see that this odds ratio could be as low as 0.37 or as high as 1.75, and hence we cannot be certain about this protective effect. In contrast, when considering peanut allergy, our estimates are greater than 1, suggesting a greater odds of allergy persistence amongst those with vitamin D deficiency. Once again, our 95% confidence intervals are inclusive of a null value, and hence we cannot be certain about the direction of effect. I would like to add that when further adjusting for eczema and seasonal blood measurement as a sensitivity analysis, our results did not change substantially. Of interest, however, when considering quintiles of vitamin D, rather than dichotomizing this continuous measurement at the cost of a greater loss of statistical power, some evidence of an association was found whereby higher levels of vitamin D corresponded with an increased odds of persistent egg allergy, concurring with the results of our primary analysis. Here, the middle quintile was used as a reference level due to a previously postulated theory in the literature of there being a U-shaped association, whereby both low and high levels of vitamin D might be associated with food allergy. Now, in terms of study strengths, the Health Nut study is valuable given its population-based nature, but perhaps more pertinently, its use of gold standard methods for detecting food allergy and obtaining vitamin D measurements. Additionally, our analysis benefited from the inclusion of confounders, thereby controlling for some potential key sources of variability. It is evident, however, that our analysis was not without its limitations. It is imperative to state that missing data and loss to follow-up were key issues. For instance, despite being a very quite large study cohort, only those with both a vitamin D deficiency and a food allergy would have even been considered for inclusion in our analysis models. And although it had been previously reported that 3.1 and 8.9% of a cohort had a peanut and egg allergy respectively, this was not reflected in our sample sizes. On account of some slight loss to follow-up, such as from those whose allergies might have naturally resolved, or simply from children refusing to have blood drawn, missing outcome and covariate values would have led to exclusion from our primary analysis model. Another issue in our study, which is part of a wider problem in the literature, is that while vitamin D levels have been ascertained, its bioavailability has not been considered. Given the role for genetic factors to influence this, such as through polymorphisms in the vitamin D binding protein, future studies should consider incorporating such information if possible. And finally, there is potential for a degree of misclassification bias when categorizing participants as being vitamin D deficient or sufficient. Despite the value of 50 nanomoles per litre holding clinical value, it is known that vitamin D levels may fluctuate on a day-to-day basis, and hence there is potential that some of those participants who we categorized as being sufficient might have actually been deficient and vice versa. A future study might perhaps consider taking multiple blood samples and calculating a mean vitamin D concentration, although admittedly, in the context of a large-scale study, this might not be so pragmatic. Overall, our analysis did not find strong evidence of an association between vitamin D deficiency at age 12 months and the persistency of food allergy by age 6 years. However, our study was inherently limited due to insufficient data. A future study on this same topic might perhaps consider recruiting only a large number of participants with pre-existing food allergy in order to better understand this relationship. Thank you.
Thank you. You are on time. So some questions from Slack. Would sex be a confounding variable in this analysis?
So previous studies on the health nut study cohort have shown that sex is generally not a factor to be a confounder. And although there is some evidence to suggest that boys might be more inclined towards certain allergies, however, in this case, we've decided that it wasn't a contributing factor in terms of vitamin D levels to be considered as a common cause confounder for adjustment.
In your DAG, you mentioned a variable maternal place of birth. Could you just talk us through that a little bit?
Yeah, so this is a proxy for ethnicity, and we use maternal place of birth as this proxy since we didn't have, I guess, a kind of measurement of ethnicity. And we've done this as it is known that the associations between vitamin D and food allergy might differ by ethnicity. An earlier study on the health nuts study cohort showed a differing association between those with parents born in Australia and those with parents born in Far East Asia.
Great, thank you very much. So thank you, Kevin, and we will move on to the next speakers. Of course, questions could be posted to Slack for you to answer. Our next speaker is Pei Xuan. Are you here? Pei Xuan is also from the University of Melbourne. You will have 5 minutes, and I'll give you a warning at 1. So if you have any questions for Pei Xuan, please drop it in Slack because we are not going to have a question and answer section after this. So please share your screen, Pei Xuan.
Yeah, can you see my screen?
Great, thank you. Please begin when you're ready.
Yeah. Okay. Thanks, Kevin, for the introduction. To save some time, I'll jump straight into the point. So as some of you may know, the cardiovascular disease, also known as CVD, is the #1 cause of death in the world. There are more than 4 million people affected by this disease in Australia, which means every 1 in 6 Australians lives with this disease. And people also found that CVD can disproportionately impact those who are more socioeconomic disadvantaged. As shown in the bar plot here, both males and females living in the lowest socioeconomic areas are more likely to die from CVD. And this concludes that the socioeconomic inequalities in CVD are apparent. The recent studies suggest that lower SCP is independently associated with unfavorable metabolomic and inflammatory profiles, which both predict CVD risks in adults. Although the adult's metabolomic profile can be predicted at age 7, the relationship of SCP to metabolomic profile and inflammation is generally poorly characterized, especially in early life. So, in addition to that, atherosclerosis, which is the main underlying cause of CVD, was found in the arteries of kids aged 10, and suggesting the development of CVD starts in childhood and accumulates risks over time. So we think identifying possible biological mechanisms in childhood that contributes to the CVD development can open up new opportunities for earlier intervention. So it brings us to our two research questions to investigate the association between SCP, metabolomic and inflammatory profile, and adverse cardiovascular phenotypes in early and mid-childhood. In our study, our data were available from Australian children recruited to the Bawang Infant Study and Children Health Checkpoint Study. In both cohorts, the neighborhood SCP and household SCP, which were indicated by CIFAR and maternal education level respectively, were measured at birth. Then the metabolomic profile and the inflammation markers and our 3 preclinical cardiovascular phenotypes, which measures blood pressure, arterial thickness, and arterial stiffness, were measured at age 4 years in B's cohort and 11 to 12 years in the Children Health Checkpoint cohort. Then the simple linear regression models were used to describe the association between each SCP and metabolomic measures. The effect of metabolomic measures on each preclinical cardiovascular phenotypes were estimated using our primary linear regression model to adjust for age, sex, and BMI. Then we have fish consumption and fish oil supplements additionally adjusted in the secondary linear regression model. And this was in light of previous studies that fish consumption could affect some metabolites concentrations and reduce CVD risks. Then our results show that lower socioeconomic position was found to be associated with, for example, higher amino acids, lower DHA and ketone bodies, and a higher level of inflammation at 11 to 12 years, which is indicated in the orange in the plot. But there was very limited evidence of an association between lower SCP and an adverse metabolomic profile at 4 years, which is indicated in dark blue. Apart from that, there were additional associations between specific metabolites, for example, higher citrate and creatinine, and increased CIMT at 11 to 12 years. Although there was an association between a higher level of glycoprotein acetyl, which is inflammation, and decreased CIMT, these findings were inconsistent with previous studies in adults. Then in terms of the mean arterial pressure, there was evidence for an association between a higher level of inflammation indicated by both inflammatory markers in our study and a higher mean arterial pressure at both of these ages. Then unfortunately for the third preclinical phenotype of pulse wave velocity, there was no association observed. So to sum up, we conclude that socioeconomic disadvantage was associated with inflammation and adverse metabolomic profiles, especially at 11 to 12 years. Then inflammation, which plays an important role in atherosclerosis, was associated with adverse cardiovascular phenotypes in childhood. More specifically, there was weak evidence for a relationship between a few metabolites and CIMT, and the mean arterial pressure increased along with inflammation in children aged 11 to 12. Then, as mentioned before, no association were observed between inflammation, metabolomic profile, and post-wave velocity at these stages. So nonetheless, we think further studies on understanding the causal drivers of these relationships may inform intervention opportunities in childhood to reduce the CVD risks. And thanks for your listening.
Thank you, Patient. That's a very interesting talk. So please, again, ask questions on Slack if you can. Our next speaker is Pavel from the Florey Institute for Neuroscience and Mental Health. Pavel, are you here?
Yep, I can't turn on my video though. It says...
That's okay. Please do share your screen though for your slides.
Okay. Here we go. Share. All right, can you see that?
Yes, I can.
All right, so hello everyone, I'm Pav, and first a crash course in alleles. An allele is a version of a DNA sequence at a given genomic location. A person inherits two alleles, one from each parent, and if the two alleles are the same, the individual is homozygous for that allele. If the alleles are different, the individual is heterozygous. A single nucleotide polymorphism, like RS5174, is like a mutant allele. The most common alleles are called wild type, and SNPs are variations to these alleles. The expression or effect of the allele can be additive, so two alleles has a stronger expression than one, and one more than zero, and dominant, where either one or two alleles is enough for expression, or recessive, so both alleles must be present for an expression. And skip forward, there we go. Alzheimer's disease accounts for 60 to 70% of dementia. A common hypothesis for the progression of Alzheimer's disease is that high levels of amyloid beta protein in the brain bind and form together as plaques, as well as an accumulation of tau, and that somehow causes neuronal cell death, but we're not sure how. So what we're interested in is neurodegeneration, and specifically ferroptosis, which is a mechanism of neuronal cell death. Now, rather than going through everything, I'm just going to say that experiments by our lab gurus led us to hypothesize that the subjects with the RS5174 allele have an increased abundance of ApoE protein, which stops ferroptosis and delays the neuronal damage typical of Alzheimer's dementia, and consequently reduces cognitive decline measurably. Of our three questions, research questions, I'm only going to focus on number one. Among those that are pathology positive, but without clinical Alzheimer's dementia diagnosis, does the RS5174 SNP reduce the decline of cognition? We're using the ADNI cohort in the first question, which had around about 414 participants. The variables of interest include the allele, and this can be measured, as I mentioned previously, in three levels, that either additive, dominant, or recessive. Our outcome variable was the clinical dementia rating scale, and our covariates were age at baseline, sex, and ApoE4, which are all involved with Alzheimer's dementia. And finally, we measured across time. To answer our research question, we ran a linear mixed model with random intercepts and an unstructured covariance matrix. The CDRSB is positively skewed in our sample. So this was transformed using a square root transformation. But in addition, so that we would rely less on the assumption of normality, we bootstrapped the model 1,000 times using a parametric bootstrap. And this is our model here. We're looking at the effect of an interaction between time in years and RS5174. These are our results. So we can see in the transformed model that the recessive model here shows a reduction in in cognitive decline. And this is my preferred way of looking at it with the bootstrap model, because it looks at the raw score. We see that it there is a reduction in clinical decline by about 1/4 of a point in the CDRSB per year. So this is very important that it's by year. So over a 10-year period, this is quite substantial. You can see that here in our little plot, the recessive here, that with 2 copies of the allele, there is a reduction in cognitive decline on the CDRSB score. And
you have 1 minute.
Thank you. As a summary, we can estimate that the effect of RS5174 is protective, ranging from 0.35 to 0.14 of a point in the CDRSB. What we haven't shown is that the phenotypic expression of the gene is recessive, but we did show that in the project, and that the effect can be replicated. And you can actually see that here. We did a meta-analysis over those 3 trials. So we had those 3 cohorts, the ADNI, ABLE, and the ADNI here, ABLE, and the BioFinder cohorts all showed a decline reduction in cognitive decline over time. Whew, 5 minutes is not a lot. And thank you to everyone.
Thank you, Powell. And you have your pet dog there, I presume?
Yeah, my wife and my pet dog, they helped me through my master's.
Lovely. Okay, thank you very much, Powell. Then our next speaker is Michael Walker. Are you here, Michael?
Yes, I am. I'll put my video on.
Yes,
Right,
and most
sharing.
importantly, please share your screen
Yep.
for your slides. Okay, great.
Okay.
So please begin when you're ready. I will give you warning at with 1 minute remaining.
Okay, thank you. So hello everyone, my name is Michael Walker. I'd like to share with you today some work I did for my MPhil in bioinformatics at the University of Melbourne a few years ago. The work was based on the cohort asthma study. This was a longitudinal study containing data on pretty much every correlated factor to do with childhood asthma known to man. Important ones were IgE or antibody blood concentrations, also numbers and types of respiratory infection, especially whether those infections were febrile or wheezy or not, or whether they were viral, and if viral, which virus was it. That data was taken year by year for the first 5 years of life, and there was perinatal data as well. So we're using IgE titers and numbers of infections and so on to predict whether a child would develop asthma. But it occurred to me, are we really predicting asthma in general, or are we really just predicting atopic or allergic asthma? Given that some of our predictors are actually antibodies in the blood, that would seem likely. Or is it just airborne atopy and so on? So how can we tell? We can't assume it, and not every predictive variable has that sort of intuitive link anyway. So what I did was based on the area under curve. I won't define it, but it basically measures the ability to sort case from control. So if your model is no better than random guessing, then it will give you a value of half. And if it works perfectly, you'll get a value of 1. In practice, it's usually somewhere in between. So if your predictive observable, say antibody titer, is truly predictive, but only of a subset of your observable, so in this case, outcomes, so in this case, atopic asthma as opposed to general asthma, then other cases, such in this case, it'd be non-atopic asthma in this example, are really only being guessed at randomly. So their contribution to the overall area under curve is only a half. So putting that together, the area under curve for your general outcome, asthma in this case, is going to be a weighted sum of the area under curve for your special case that you're actually predicting, and half for random guessing. Rearranging that weighted sum, we get this weighted difference, which I've called the exclusivity index. So its properties are, first of all, that if your predictor is truly exclusive, it's really only predicting a particular subset, then its value is half. Otherwise, its value is just equal to the area under curve of your outcome. So just here are some examples in this table. So we're looking at the predictivity of house dust mite titers in the blood in the first 2 years of life. So the top 2 rows there, and we see that the exclusivity index is within error of half. So that means that when we think we're predicting wheeze, we're really only predicting atopic wheeze. In fact, we're only really predicting aeroatopic wheeze. So that's atopic wheeze where you're sensitive to an airborne allergen. Going down to the peanut IgE, so the peanut allergy, again, if you think you're predicting wheeze, which was our proxy for asthma, you're really only predicting atopic wheeze, so allergic asthma. but not airborne atopy, and that's to be expected because peanuts are, a food. Atopy number there is also exclusive. That refers to the number of allergens to which you might be sensitive. Importantly, the bottom two rows, severe and febrile lower respiratory infection, even though they're known to be good predictors for asthma or for fifth year wheeze, they are not exclusive, so that they don't predict a particular type of asthma there. This table is the
I have
equivalent.
1 minute left.
Okay,
Michael.
thank you. This table is the equivalent thing for non-atopic asthma. So the number of children in the home, number of older children in the home in the first few years of life is known to be protective against asthma. But if we have a look at the exclusivity index before it, we see that actually it's within error of a half. So it's only protecting you against non-atopic asthma. Your probability of getting atopic asthma is not affected by older children in your house when you're born. Again, going back down to the severe and febrile lower respiratory infections, the bottom two rows there. Again, known to be good predictors for 50-year wheeze for asthma, but nothing to indicate that they are predicting this exclusively. So that it appears to be a very general predictor for asthma in general. HRV stands for human rhinovirus. Why infections of that would predict non-atopic asthma, I honestly don't know, but it does appear to be exclusive for that also. I'll end it there. So thank you everyone for your attention, and I'll take any questions if anyone has any.
Thank you, Michael. I believe we are leaving the questions to Slack. So if you do have questions to any of our speakers of these sessions, please leave those questions in Slack and they will answer them in a thread. So with that, I would like to conclude this session and thank all the speakers, exceptional, really. And that we will have some time for break and come back for session 6.
Awesome work. Thank you very much, everyone. Thanks, Kevin, for sharing this session. Yes, we have 7 minutes. We are on track pretty much. So great job, Chair, catching up, and also Summer, cutting and keeping the discussion there. So I think we can have 7 minutes break for this one. Thanks, everyone.